Welcome everybody to our seminar on the next generation of plant-based meat analogues. I'm Matt Stading. I'm a professor in material science at Rice. I'm also project leader for this project and will be the host during this seminar. The project is a cooperation between Lantmännen and Orkla, and it's partly funded by the Swedish innovation agency Vinnova. Today we will share the findings, but before the start I would like to point out that we will record the presentations, not the questions and answers, but we will post the presentations later on our website. Emily Dybeck will help handling the chat questions, so if you have questions to the seminars, please place them there and we'll try to answer them either today or later on. So the background for this project is, as you all know, that meat contributes strongly to global warming, and excess intake of red meat and processed meat is bad for health. You have probably seen this diagram before of emissions of greenhouse gases, and we can just conclude that the plant-based foods is to the left and the animal-based is to the right. So if we look more on sustainability, the food chain in itself contributes with about 25% of the total climate gas emissions, and meat production is actually more than half, about 14% of the total greenhouse gas emissions. So this is really a strong motivation for doing something else than meat. And also, if we would reduce meat consumption, it would have rather a drastic and rapid effect on global warming, as methane is an important factor, and that has a relatively short half-life in the atmosphere. Another factor is that the arable land is limited, and we still use about 70% in the Western countries for production of feed. And if we look at health effects, we consume rather a different amount of meat in different countries. The world average is about 40 kilos, and we are about twice that in Sweden and also in the rest of Europe. There has been a European goal of about 40 kilos, so that is about half, and the recent Nordic nutrition recommendations says about 350 grams red meat per week, so about 18 kilos per year, so that is even lower. And we have actually doubled the meat consumption in the last 50 years, and the ones of us that were present or kids in the 50 years ago, I don't think we felt deprived of meat, so it shouldn't be that difficult to decrease current consumption to half. It's well established that red meat and processed meat is not good for health, but it's also a fact that meat is an important food and has very big nutritional advantages. So it's a good source for protein and also for iron, for example. So it's fair to say that all of us should not reduce meat to the same amount. So children and seniors and women in fertile ages should not reduce the meat consumption as much as I do and other men in my age. We also know that there are alternatives like plant-based meat analogues, but the iron availability is rather limited, and we will come back to that more later on. Yeah, so what could we do? Well, we could stop eating meat and just eating plant-based food instead, and that would be perfectly fine. But because we would get the essential amino acids by a mix of cereals and legumes that has been well established during thousands of years, and in this way also the arable land would be sufficient to feed the expected global population of 12 billion people. Another way is to use other sources like insects, and when I present this to a live audience, I also see people frowning a bit, and because it is a big cultural threshold to eat insects. It would be good because it's easy to produce and the greenhouse gas emissions are low, for example, but it's also difficult to make a business case out of it because the extraction of protein is rather costly compared to the growing. We could, of course, also think about growing meat in the lab or in the factory by tissue culture. And then you take muscle cells, you grow them on a scaffold, and that means that you have an animal-free alternative to meat, but it's still muscle cells. I think that the consumer acceptance is a bit limited, but not too bad. And it's quite a hot area, and it's a lot of companies are working on this to scale it up. And if you look at the picture to the right, that's from a previous project where we cultivated muscle cells on a scaffold of plant proteins. So the majority of the mass was actually plant proteins, and then we had sort of muscles on the, like the icing on the cake, to give it a bigger, better meat feel of the final product. But it's not that straightforward. I mean, it is good because you have lower greenhouse gas emissions, but it's more energy intensive. And just because it's muscle cells, it's not really meat, because meat is fat and it's connective tissue and it has a higher structure than just muscle cells. Also, the tissue culture medium has to be developed. If you do it in the lab, you use calf serum, which is not very sustainable. And maybe most important is that the production has to be totally sterile, because bacteria grows equally well as cells during these conditions. And if you look at the picture to the right, it's from one of the pioneers in the area, Mark Post. He scaled up just by making hundreds of Petri dishes. And then in the final part of that project, they made a hamburger, and that was presented at a big event. It was rather costly, so this is not the way to scale up, but this is not how companies are working today either. Well, extrusion of plant-based proteins is what we have been working with in this project, and it is an efficient production. You can use materials that are available in large amounts, like soy, pea, and wheat protein. The process is fairly straightforward. You mix protein powder and water, then you mix it in with the 2 screws, you heat it to about 140 degrees, then the protein melts, and when you push it through a cooling die, fibers are spontaneously formed, and you end up with a structure that is similar to chicken meat. There are nutritional concerns, as mentioned before, because you have decreased iron bioavailability, and you also have too many oligosaccharides. So that's why you can use fermentation, as we have done in this project. So finally, what have we focused on in this? This is rather a general area, but what the Like Meat project has focused on is firstly that we have used fermentation and enzyme treatment on the raw materials, which means that we can decrease phytic acid and oligosaccharides quite a lot. We also have investigated the functional properties, how the fermentation affects the functional properties. We have used extrusion, as said, and then we have also tried to understand the fiber formation mechanisms. It's rather straightforward to set it up so you get fibers, but it's not really known why the fibers are formed, so that requires quite more work. We have investigated optical formulation and also a processing window. We have used mainly protein concentrates, which are believed to be more sustainable. We will hear more about that too, because the process is simpler than to produce isolates. But isolates have sufficient protein content, which the concentrates don't for this type of extrusion. And then we have tried to answer the question, for example, can protein concentrates be used instead of isolates, and other similar questions. So with this said, we will go into more details, and then I leave the word to my colleagues. And we will start by looking at the fermentation, and Dr. Karin Bjerre will present the benefits of using fermentation on legumes. So please, Karin, whenever you're ready, the floor is yours.
Well, good morning, everyone. I'm going to talk about the fermentation part in this work. Humans have used fermentations for thousands of years to preserve food, and we still use that technique. In your own home, you probably have several of these foods at home. For instance, beverages like wine and beer, bread, vegetables, yogurt, salami, or cheese. Milk is a very good example. If you have fresh milk at home, you can store it for a week. If you have fermentation to produce a yogurt, it can be stored for about a month. And if you do cheese, the nutrients can be stored in a safe and efficient way for about a year. So fermentation is used to increase the shelf life, to change the taste, reduce antinutrients, increase the product safety, change the texture, and improve nutrition. And some commonly used microbes are lactic acid bacteria, for instance in yogurt, cheese, vegetables, and salami. The microbe produce lactic acid and then lower the pH, which makes the food safe. Saccharomyces cerevisiae, baker's yeast, is also commonly used for wine and beer production and also for bread. The yeast produce ethanol that preserve the beverages and also produce carbon dioxide that produce this fluffiness in the bread. You can also use fungi for taste and texture, for instance in cheese, some Asian products like miso and tempeh. So how does it work? Well, you have a small microbe that wants to divide and produce more microbes. And to be able to do that, the microbe needs food. It needs nutrients, for instance, sugar, nitrogen, and other trace elements. And if the microbe can grow, it produces biomass, gas, ethanol, acids, and other products. But if you have a specific question, if you have a specific task that you want your microbe to do, you need to look into the literature and you need to look at your microbe. What kind of genetic potential does the microbe have and what kind of metabolism can the microbe do, and select the right microbe for the right task. In this project, we have used pea concentrate for fermentation and tried to optimize the process for process parameters for extrusion, but also looking for nutritional values. And there are antinutrients in pea. I'm sorry, this one is in Swedish, but as you can see, if you look at the pea, ärta, you do have oligosaccharides, and there are also phytate present. And both of them are considered to be antinutrients. And can you reduce this by fermentation or enzymes? Yes, if you look into the literature, for instance, with oligosaccharides reduction, you can use aspergillus, which is a fungi. You can use lactic acid bacteria and some commercial enzymes. There's also plenty of phytic acid in plants, and this is the major phosphorus storage in many plant tissues. But it also binds iron. And this is very important, especially for vegetarians, since the iron in the plant might not be available for absorption in the gut. And it has been reported that the phytate iron molar ratio should be below 1 or preferably below 0.4. And this is how the phytic acid molecule looks like. It has 6 positions for phosphorus, and they can bind to metal ions, for instance, iron. And this is quite stable. So you would like these phosphorus groups to be cleaved off, and by that making the iron more available for absorption in the gut. For instance, in pig feed, they regularly add a phytase that cleaves these bonds, and that's very good for in pig feed for growth for the pigs. And if you look into the literature, there's good hope. You can use enzymes, you can use actually extrusion to reduce the phytate. There's several microorganisms reported to do this job as well. My lab in 2020, she showed that Pediococcus could reduce phytate in faba beans, and also been shown that some lactic acid bacteria and some aspergillus can also do that. So what have we done in this project? We started with a screening in small scale. We used several different bacteria, some yeast and fungus, to see if they could grow on this pea concentrate. So we just looked at growth, appearance, and smell. Could they use this food for growth? Could the microbes use these nutrients available in the pea concentrate? And based on these results, we selected 2 microorganisms to work more with. We chose 1 lactobacillus and 1 fungus. We also tried a few enzymes, a phytase, and a few different amylases, and we looked at pH and appearance. And after fermentation, we took samples for starch, oligosaccharides, phytate, amino acids, proteins, and protein degradation, water holding capacity, and water solubility index. We also tried to optimize the process due to viscosity, time, and temperatures, and amylase. And we combined different enzymes and bacteria. And the final part was the pilot scale experiment, because if you want to do extrusion, you need a few kilos of dried material. So we chose a process of enzyme treatment with an amylase and a phytase, and 1 lactobacillus for fermentation. And after the fermentation, the fermentate was spray dried and used for extrusion. We have been sending out samples for analytics and microbial samples for this, but they are not. We don't have these results yet from this part. Here are some results for the more small-scale fermentations. Here are results from the fermentation with 1 lactobacillus and the fungus. And you can see the reference. We have the 2 oligosaccharides, stachyose and raffinose. And after lactobacillus and fungus fermentation, you can see that both microorganisms can do the job in reducing the oligosaccharides. And this is duplicate fermentations. And oligosaccharides can give you abdominal pain. Phytate we investigated as well, and here you can see results, the content of IP6 in P concentrate after enzyme treatment, and we also tried the yeast, a bacteria, a fungus, and lactobacillus. And after this treatment and fermentations, you can see that the phytase treatment reduced IP6 to 0, and it was below detection limit. And so the reduction was 100%. The yeast we selected can only reduce the IP6 by 10%. One of the bacteria can reduce it about 80%. The fungus about 50-60%, and the lactobacillus about 8%. So the only treatment that could get to the level of below 1 or 0.4 was the phytase treatment. We have also been looking at proteins, protein levels, and protein degradation, and here is one example of that. Here is an SDH page, a gel, where you run the proteins on a gel and can look at sizes. So there are 3 standards in this picture, and if you look from the right, you can see the P concentrate in triplicates. This one isolate on this gel as well, and then you can see after phytase treatment and after lactobacillus fermentation. And after the lactobacillus fermentation, you can see that some of the proteins have degraded, and we also have some extra bands. We have also measured starch, and here is one example of starch. We have a reference and duplicates of fermentations after lactobacillus and fungus fermentations, and as you can see, the starch levels have been reduced after these treatments. We have also been looking at microbial safety for the lactobacillus and the fungal fermentation. Before and after fermentation, we could not see any listeria or salmonella. We have also been looking at total count. Aerobic bacteria is what was about log 3. We could detect some spores, and we have looked if we can find any Bacillus cereus, but we could not detect that. And we have also seen that we have about log 2 of yeasts. We will also look at microbial safety from this pilot scale fermentation, and also look at how it looks like in the extrudate. And in the final part, in the large scale, we performed 1 pilot scale fermentation at Balsgård near Kristianstad. And we selected 1 process with an amylase and a phytase, and then we added 1 lactobacillus. And here you can see the pH measurements from the fermentation, and as you can see, it has a bit of a lag phase before the reduction of pH starts. And then we have a very nice pH reduction, which means that lactobacillus are eating the nutrients in the slurry and lowers the pH by producing lactic acid. At the end of the fermentation, we took out the fermentate and stored it in buckets in cold. Before spray drying, the pH was adjusted so it resembled the peak concentrate. So this was from the fermentation part, and I thank you for your attention.
Thank you very much, Karin. And then we move to another part, which is about the extrusion that we have performed in the project. And then we'll introduce our extrusion expert, which is Leila Dahl, who will share their findings from the practical extrusion with you. So Leila, please, whenever you're ready, you can share your screen.
Right. So now you see my screen and you can hear me. Yes. So hi and welcome everyone. My name is Leila and I work at Rice Food and Agriculture, where my main focus is on structure and prototype engineering. And I work a lot with extrusion technology. So today I will present the work that we have done in the Like Meat project on extrusion of healthy and sustainable meat analogues. Right, so here you see an overview of what I'm going to talk about today. So we will start with a short introduction to the extrusion process and what we mean when we say high moisture meat analogues or HMMAs. We then move on to look into the inline analysis of protein extrusion, and this with regards to the practical work that have been done for further simulation. We also looked into the protein content within extrusion to try to find sort of critical limits where we no longer get the achieve the structure we want or the process stability. So the fermented samples that Karin talked about earlier were also extruded, and we will share our learnings about that. And then we'll also talk a little bit about how the functionality is affected by the fermentation and the enzymatic treatment. Right, so a brief introduction to extrusion. It is a well-established process for producing foods such as pasta, snack foods, or pet foods. However, in recent years, a lot of focus has been put on how we can produce meat replacers using the technology, and this based on plant-based proteins. So the extrusion process will expose the material to mixing, heating, shearing, and pressure. So material is fed in the early zones, and then it travels along the screws and eventually enters a die. And you can vary the temperature in the different zones of the barrel. In the case of HMMAs, the material will travel through a cooling die, which will create a flow profile aligning the fibers. In the image here, you can see an example of a HMMA, and you can see how the fibers are aligned. So compared to a dry extruded meat analogues, these are produced with a low moisture content and a smaller opening at the die. And when the material leaves the extruder, they will expand and you get more of a spongy texture. These types of products also need to be rehydrated and shaped into the desired products or packed up into a mince. Whereas for HMMA, you can create a structure more similar to meat and where you can have whole chunks. Right, so there There are still many unanswered questions as to how the fibrous texture in an HMMA is created in the extrusion process and what the critical factors are. So to be able to deeper understand these mechanisms, various approaches to inline analysis has been taken in this project. So we have used ultrasound sensors to be able to measure the flow in the dye. And for this, a specialized dye was designed. We also looked at the influence of the cooling dye length and the temperature on the rheological properties of the material. To evaluate the influence of water and protein content on the rheological properties, materials were produced with different protein content and moisture content and collected right before the stage where they would normally enter the dye. Colorants were used to visualize the flow profile in the extrudate, and this by adding the colorant with the dry material and then collecting the sample as it leaves the dye. The generated data was then used for simulating and understanding the fiber formation mechanism. So extrusion trials were performed with two main purposes. So first, to investigate the influence of the protein content and where the critical limits can be found. Also, in relation to this, we looked at the synergies and differences between concentrates and isolates. So concentrates, as mentioned, are produced in a dry process, which is likely less energy intensive than isolate production and therefore better from a sustainability point of view. However, the resulting materials will differ in both the protein content and the functionality of the proteins. So concentrates are generally less favorable for a high moisture extrusion process of meat analogues. The second purpose was also to investigate the potential of extruding fermented materials. And fermentation will, of course, influence several factors. So the components may be degraded or some components may be produced, which will influence the composition, the molecular weights of the different components, and so on. So prior to the fermentation with lactic acid bacteria, enzymatic treatment was performed to reduce the starch and the phytic acid content. Yellow pea protein was selected as the plant protein to investigate, and this because it may be produced in Sweden with an already established cultivation. And for initial trials of evaluating the extrudability of fermented materials, 2 commercial fermented pea protein isolates were used. No fermented concentrates could be found on the market. Right, so here you see an overview of the work done to investigate the influence of the protein content, the difference between concentrates and isolates, and extrusion of commercial fermented pea isolates. So on the left side here, you can see the work with the pea protein isolate, which was either combined with pea fiber or pea protein concentrate. A green marking means that we have sufficient texture and a stable flow, while the yellow and red markings indicate that we did not achieve this. So the main conclusion drawn for the study of protein content was that the level should be kept over about 60%, and while still being able to achieve a stable process and a good texture. And this was, of course, for the materials and the combinations evaluated here. And when I say process stability or instability, I mainly refer to the feeding of the material and the stability of the flow through the extruder and the cooling die. So what you often see during instabilities is that the material in the inner part of the extrudate will flow faster than the outer, and you don't get a coherent structure throughout the whole material. Inconsistent feeding will also result in a pressure fluctuation and may be caused by the powder properties. So this moves us on to the synergies and differences between isolates and concentrates. So P concentrates are generally very sticky and they tend to clog the hopper, which is highly influencing the feeding. Another significant functional disadvantage of the concentrates is their low water holding capacity. This means that you are not able to incorporate as much water into the product, and it may also cause problems with backflow, meaning that the material is not able to hold the water and transport it as well through the extruder and might go backwards. To the right here, you can see the work with the fermented commercial isolates. And there we tried 2 different materials. And overall, the extrudates for these were softer than the ones with the non-fermented isolate. However, the differences between them was very big, where one was significantly softer than the other. Right, so we will now look into the extrusion of the pea protein concentrates fermented in this project. And yes, here you see a brief overview. So we focused on the spray dried material, as this is a more relevant drying technique industrially than freeze drying. And we extruded both the final fermented material as well as the material that had been exposed to the enzymatic treatments to be able to see the differences between the different steps. As a reference, we used the starting material without any pre-treatments. Yes, and the first extrudate to your left here is of the non-treated starting material. And as you can see, there is none or very little tendencies to a fibrous structure here. The flow and the process was also very unstable. So after the enzymatic treatment, we can see that the process stability was highly improved, which can be partly linked to the feeding of the material. However, we cannot see any improvement in the structure here. It still has no elasticity and it falls apart quite easily. But further, after fermentation, we have an improved elasticity and a nicer texture, more fibrous. So something has happened. And the results can be, as I said, partly explained by the improved feeding of the material, but also the composition should be altered by the fermentation, where, for example, the starch has been degraded, and the presence of starch could otherwise negatively interact with the fibrous formation in HMMAs. We also have the influence of the functional properties, and here we look at the solubility and the water holding capacity. So as I said, water holding capacity is an important property when it comes to extrusion, and concentrates generally have a significantly lower water holding than the concentrates. This means that you can't put as much water in and you can have problems in the process. What we do see after the fermentation and the enzymatic treatment is that we have a somewhat higher water holding capacity, which indicates an improved extrudability. The water solubility is related to the water holding and tends to go down as the water holding goes up. And that is what we see here, that it has slightly gone down after all the different types of treatments. Yes, so with that, I want to thank you so much for listening. And if you have any questions, don't hesitate to ask. You can also contact me afterwards at my email address written here.
Thanks very much, Leila, for very clear and thorough presentations. Just to note that in the introduction, I mentioned that extrusion is fairly straightforward. That doesn't mean that it's easy, as you've seen in the previous talk. And it also doesn't mean that the interpretation of what you get out is easy. And now we will elaborate a little bit more on that. And that will be a presentation by our simulation expert, who is Dr. Erik Kaunisto, who will present more on the interpretation of the results from the extrusion. So Erik, please go ahead with your talk. The floor is yours.
Thank you, Mats. Thank you for the kind introduction. Do you all hear me and do you see the screen? Yes, good. Okay, so this talk will be about the theoretical and experimental part and characterization of fiber formation in P dye-melt flow during extrusion. And with dye-melt here, I mostly refer to a blend of water and P isolate and some small amount of fibers as well. And the goal with this talk will be to try to elucidate how fibers are formed when extruding this piece. And one could say that this is a rather abstract topic. It quickly gets pretty nitty-gritty. And my ambition here is anyway to try to give an overview and be as tangible as possible about these things, and really try to give an idea on how our current know-how within this field has developed, and what the present state of the art, so to speak, is within understanding what happens during extrusion. So with that said, let us proceed. I will first give an outline of the talk. And first of all, I would like just to, as Mats and also Leila has mentioned, give a short overview of the extrusion process, which in this case is for high moisture meat analogues, which is abbreviated, as has been said, HMMA. And then one could arguably ask, why should one care to model fiber formation anyway? Why can't we just make experiments? And I would try to give an answer to that question. And given the answer to that question, there is a need to characterize the flow properties, which is mostly the viscosity in this case of P's, or this P melt. And then I will go into how we have modeled the flow in the extruded die, and also the fiber formation that actually occur in the extruded die. What kind of model do we have to describe that? And based on these models, I will present some results and conclusions. So first of all, the overview. As has been mentioned here, we have an extrusion process for HMMA, and we have a blend of water and protein, mainly protein isolate, that goes into the extruder in this case. And then we have a transport of this powder and water. It forms a melt when subjected to shear forces, and then the melt is finally pumped through the final stages of the screw into the cooling die. And this is essentially where, within quotation marks, the magic happens, where we actually have the fiber formation mechanism. But we have a preconditioning of the proteins in the actual barrel section and the screw sections. So when it finally enters the die, we have the cooling, and that's when fibers are formed and aligned as a combination of the temperature profile in the die, and also the shear forces that are present, and the strain rates that are present. And if we do this correctly, we should have a fibrous meat analog in the end. So just to give some incentives then why we should model fiber formation, well, we can fairly say that we don't really know how fibers are formed. There's a lot of discussion in the literature about this now, and there are lots of hypotheses as well. And it's starting a little bit to converge, but it's still very open. And exactly how you should refer to a fiber and exactly what constitutes a fiber is a little bit of a semantic discussion as well. But anyway, the main problems that we face is essentially related to efficient scale up of the extrusion process. And also that if we don't know how these things are working, we really can't utilize efficiently both existing and novel protein sources in a good way when we want to scale up. And a main problem here is that the literature and the research field here is quite young. There's been some published, but it's as I said, there are lots of opinions and lots of models that try to explain this. So there's a lot to do still in the literature, and there's really a shortage of a comprehensive model that really explains this from sort of what happens when you insert everything into the extruder and then what really comes out and the whole process itself. So it's sort of a black box, but it's getting more white. And I would say since the pioneering work done by Murillo in 2019, it has actually been shown that something that resembles phase separation may actually be partially an explanation to why we get fibers from the melt in the cooling die. And there are some molecular mechanisms that are behind this. Even if we call it phase separation, it could possibly be more of a syneresis, but that could arguably also be, it's a little bit of semantics here, what you call a phase separation. But anyway, these are caused by cross-linking and the molecular level dynamics that are important for fiber formation. And I think based on a recent literature review, the phase separation hypothesis has definitely been included. So the current aim here has been to further investigate that for realistic extrusion conditions, again, then using P isolates and water as a model substrate. So the first thing we did in order to model this was to make a flow characterization or a rheological characterization of the material. And that is to estimate the flow properties, both as a function of shear and also about temperature, to really cover the conditions in the extruder, both in terms of the shear rates that are present, but also in terms of the temperatures that are present. So we have a mapping of all the conditions to be utilized in the model. And the rheology that we have measured is trying to mimic intentionally the one we have at the cooling die inlet, because that's what we easily can measure. And that's what our hypothesis for the initial fiber formation actually starts from this rheology. So it seems like a reasonable starting point to estimate the rheology of this mixture. And then we can see that the data here adheres to a power law description. Everything falls on a power law line for different temperatures over the region of strain rates that have been investigated here. And we also have a temperature dependence that seems to follow Arrhenius type model expression. So we argue that we have a fairly good empirical description here with our models for the rheological input to the model. And based on this, we have then defined a flow model. The flow model is essentially represented by the Navier-Stokes equations in a 2D representation of the cooling die, where we resolve the narrowest part of the cooling die, where we have the largest amount of shear forces for texturation. And then the flow properties in terms of viscosity are used as input to the Navier-Stokes equations. And the heat equation for the temperature that is transported in the cooling die due to the cooling at the walls of the cooling die will in turn affect the flow field through the viscosity. And all these equations then form a coupled system where temperature, flow properties, and flow affect each other, as in the picture to the right here. So I have intentionally left out any equations here. There will be other sources if you want to read upon that in more detail. But essentially, we have this very implicit coupled system, which is very hard to a priori sort of understand how it works. So you need to really simulate this connected system to really get an understanding for what happens and what kind of flow profiles, temperature profiles, and properties you get for the phase separation that occurs in the die as we believe it. So based on this flow model, we then go to the phase separation model. And the phase separation in this case is modeled by simply solving the Cahn-Hilliard equations for the separation of a two-phase system, or we could argue that it can be simplified as a two-phase system where we have a protein-rich phase and a water-rich phase, respectively. And the phase separation in this case is driven by the amount of subcooling below a critical temperature. And the input to the equations essentially are the surface tension and the interface thickness that we want to resolve, as well as the speed of phase separation. So with this definition, we essentially assume that we can call a fiber the protein-rich phase that emerges from phase separation. And as you can see in the picture here to the right, I tried to illustrate the concept of phase separation here. You have a somewhat homogeneous mixture with small disturbances. And when we subcool this system, we eventually get out structures in this case a spinodal pattern, which would resemble fibers. And these look rather chaotic, but given the shear and the flow in the die, we can have alignment of these structures into fiber-like structures, which is essentially the assumption then for what is happening in the cooling die. So with that said, we can jump to the next slide and presents the results related to the flow in the die. So as you can see in figure A, we have modeled the flow, and the flow here features essentially viscous regions close to the walls where we have very slow flow rate. And that poses a problem, both because we don't have any shear forces in these regions, and we have also we have subcooling, so we can have phase separation in this region, but we don't have much of alignment. And this is what you can see, that we have a fast flowing center, which is red, which is essentially where the system has not been cooled enough. So the viscosity in this region is pretty high, and then it's easier to flow there. And that also means that we essentially form a small transition zone in this case, where we have these higher strain rates, which are the prerequisite, as we believe it, for alignment of the formed fibers. So if we are in these blue zones, we could possibly expect phase separation, but we might not expect as big of alignment as in the case of the red zones in figure B. So this is a problem, and we really want to understand how this picture actually changes throughout the die. This is simplification, which is based on the inlet rheology of the die, but as I will explain to you later, this is not the complete picture, and we still have challenges to really explain what happens when the flow actually converges into the plug that we actually see going out of the extruded die in the case where this works as intended. I have also plotted here, as you can see, the isotherms, which would be corresponding to, for instance, an assumed critical temperature. And in that case, you can see that the phase separation will only occur within the confines of these isotherms as bounded by them and the outer walls. And the inner core, depending on this value, might be weakly to not fibrillated at all. And I will show that in the next slide. So here are some results from simulating the Cone-Hilliard equation and the phase separation in this given flow field that I just introduced. And we have done that for some different geometries and the coolings at the edges. And to the upper left, you can see that, let's put on the laser pointer here. So you can see that we have a cooling of 60 degrees, and we have a length of one modular cooling die here of 200 millimeters, and we have a width of 1 centimeter in the die here. And what you can see is essentially that we get only a random pattern essentially at the walls, but we have weak to none fiber alignment. It's only towards the middle of the core where we don't have any phase separation at all in this case, and very weak fiber formation, and essentially not at all in the middle, actually, in the green regions. And that's a problem. But when we lower the cooling temperature, we can see that the core actually shrinks, and we start to get more of alignment close to the boundaries of the core, where we can see that, for instance, the protein-rich phase, if we assume that is the red region here, then we get some kind of alignment across these borders here. But it's a very narrow region, and we don't really understand why this happens throughout the entire cross-section as we see when it works. But we believe that this is because of the changing rheology that I will get back to. And in the lower left corner, you can see also what happens if we increase the length instead of the cooling die, and we put 2 modular cooling sections instead of 1. Then we can see that the temperature has time to reach the center of the cooling, has time to reach the center of the die, and we can start getting weak fiber formation in the middle of the die, but it's essentially very lumpy, and it's not very thin fibers that are formed. So the thin fibers are only formed in the region where we have the maximal shear and maximal strain in this case. And also similarly, we can achieve a similar situation with instead decreasing the width of the die instead of increasing the length, because that's also going to improve the heat transfer and the amount of cooling that can reach into the middle of the die. And in this case, we get sort of a similar pattern with the bulky middle with weak fiber formation, and then we have closer to the walls a little bit more chaotic. And we have this region with aligned fibers close to the region of maximal strain rate in the flow. So there are similar ways to achieve this. In some sense, we realize here that length and width are somewhat interchangeable, at least in this idealized world that we present here. But it's good to know that very small changes in the die width actually can be of great significance when we're talking about the length of the extruder. So that's something that could potentially be important for an extruder die manufacturer to know about when trying to design a system for production of these products in large scale. So let's move then to the next slide. Here again, I have a similar situation, but we also know that given this smaller die, we may have a dependency on phase separation speed related to the viscosity, because the viscosity of the material changes throughout the die, especially when it's cool. The viscosity gets much higher. And given that we know that mass transfer and diffusion rates are very much affected by viscosity increases, we know that the phase separation speed should arguably be decreased when we increase the viscosity. So that would mean that if we have a connection between the viscosity and the phase separation speed, we get much smaller fibers at the edges than we would if we don't have this. So this might also be a partial explanation to why we could expect smaller fibers at the edges and more bulky fibers perhaps in the middle. Not that actually any size differences could be explained through this mechanism. The fiber consistency at all also can be plotted if we don't really care about the phases and instead start caring about the absolute value of the order parameter here, which is in some sense the measure of the concentration in the phases. Then we can see that the absolute value actually would represent a intensity of the phase separation. And that would mean that in the middle, we have less phase separation, as I've been saying, and at the edges, we have larger phase separation. And thereby, we could possibly have a relation between fiber consistency that may vary across the die width. And that would be something to take into account also when making this simulation. We want to achieve such a homogeneous fiber consistency as possible. That would definitely be a goal. So that there might exist such a correlation is quite exciting. We don't really know whether this is the case because we haven't really tested that in a sensory way. But that's at least what the model seems to suggest. So quite interesting to study further. And with that, we come to the experimental part, which Leila has already been presenting. So I would just add a little bit on the actual flow profile that we get when we measure. And this is where we actually have our discrepancy with the model at present, and also a possibility to learn something. Because what we see is not the kind of long core that we get in the simulation that comes out. We see a plug coming out when it works. And that means that we somehow have a contraction of the plug within one modular cooling die in this case. And we can see that we, of course, have this core slip boundary that in some sense are represented by the simulations. But this is occurring much faster. This contraction is occurring much faster than what is suggested by merely basing the simulations on the inlet rheology. That means that we probably need to account for the changing rheology during this simulation in the extruder, and that we need to actually have that as a new sort of input to the model to actually model this contraction in a sound way. But it's a crude approximation, at least in this region, and it gives some insights into what is happening. And we also complement this with a light microscopy image on the extruded sample. And there we can see that we actually have the flow direction, which seems slightly parabolical, which is also consistent with previously published literature. But we also see these small white cracks, which we're very curious about. And those could possibly be tears that have occurred mechanically due to the fact that these were regions that have phase separated and being water rich, which means that they don't have any structural integrity and would just crack when they dry out. So this seems to be some kind of evidence for the phase separation hypothesis, but it needs to be investigated further. And also the scale of these things is a little bit different than in the simulations also, which we have arguments for when we have published our results, of course. So it's quite tricky to get this to represent what happens here, but we have at least a beginning of an explanation for this. So given that, some conclusions. The dye inlet melt rheology provides a limited picture of the flow behavior in the dye, and it does not fully account, unfortunately, for the rheological changes suggested by the experimental flow profile. But this discrepancy is something that we can actually, as I said, learn something from and have as a ground for further studies. So it's quite exciting. And the dye width and length are somewhat interchangeable to achieve fiber formation in the model. The impact of viscosity on phase separation speed may also affect the fiber formation close to the walls, because we know that diffusion is coupled is inversely coupled, I should say, to viscosity usually. And the fiber consistency may vary across the cooling dye, but it remains also to be seen if this is really the case and if it has some predictive capabilities. The white cracks in the light microscopy images indicate potential presence of earlier water-rich domains, which would validate our hypothesis or strengthen it at least. And then future work would have to somehow, I guess, focus on either resolving individual phase viscosities in the phase separated states, or use some kind of a time spatially dependent bulk viscosity between the conditions at the exit and at the inlet, or possibly inline monitoring of these systems, as we also have tried and also as Leila described. Then there are other things that also might complicate this, and this is the fact that we may also have non-ideal boundary conditions at the walls in the extruder cooling dye, due to the fact that if phase separation syneresis, or however we want to phrase it, lets out a lot of water, that may lubricate the sort of edge, and that might lead to a slip across the edge. That's something that may have to be accounted for, but it's not apparent how to do that. So that's something we must consider in the future. And then, of course, if someone wants to read up on the details of this was just a very brief overview. Then you may want to read our paper on this that's recently been published. I think that's it. And I want to thank you all for listening. And of course, if you have any further questions regarding any modeling details, and if you feel that something was unclear, please contact me at eric.kaunisto@ri.se. So thank you.
Thank you very much, Eric. Okay, so let's move on to the next presentation, which is by Dr. Elinor Hallström. And so far we have sort of touched on how you do this, but one of the criteria to starting this project was that we want to have something that is more sustainable. So do we? Elinor has looked at this more widely than just this project. So please let us know about the climate impact in general of plant-based meat analogues, Elinor.
Yes, let's see. Can you hear me?
Yes.
Good, and see the screen. Perfect. So hi, everyone. As Mats said, now we're going to change the perspective a bit and talk about environmental impact, and more specifically about the climate impact of plant-based meat analogues. Plant-based meat analogues are often proposed as a viable lower carbon alternative on the plate, but what do we really know based on current research? My name is Elinor Hallström, and I am a researcher in sustainable nutrition at RISE. I will start off with a brief introduction. The transition to more plant-based diet is believed to be an important measure for more sustainable eating habits, especially in countries like Sweden, where we have high consumption of animal-based foods. We also know that a large part of the average Swede's climate and environmental impact from the diet comes from animal-based foods. Research estimates that about 70% of total dietary climate impact in Sweden comes from meat, dairy, seafood, and eggs. But the question then is, how can we achieve a shift towards a greater proportion of plant-based foods in our diets? And what more specifically should we eat more of? Well, one answer to this question could be that we should eat more whole grain products and legumes. A higher intake of whole grains and legumes are the dietary factors believed to have the greatest potential to improve public health in Sweden, according to the Global Burden of Disease study. And whole grains and legumes are also products having a low environmental impact compared to many other foods. But what we see in the society today is partly something else. What the consumers are looking for are often foods that resemble meat and dairy products that can be cooked and eaten in the same way and also taste in the same way. In other words, plant-based meats and dairy analogues. And the market for this type of food is, as you are well aware of, growing rapidly. However, if increased consumption of meat analogues is to contribute to more sustainable eating habits, they also need to be both nutritious and have a lower environmental impact than the animal-based products that they are supposed to replace on the plate. So, what do we then know about the climate impact of plant-based meat analogues in relation to other foods? Well, surprisingly little we realized when we started to have a closer look at this. This is clearly a growing area of research. However, existing life cycle assessments are often difficult to compare because they use different methods. And moreover, reviews and meta-analysis that compiled and compare results from life cycle assessments of foods, such as the examples given here to the right, do so far not include plant-based meat analogues. And in these studies, where also this famous quote comes from, plant-based foods are instead represented by less processed products, such as legumes, nuts, cereals, and tofu. So the limited knowledge about the climate impact of plant-based meat analogues in comparison with other foods was really the basis for why we performed our review analysis that was published this spring. The aim of this study, which was carried out within the Research Center of Finest, was to summarize and compile existing research in the field and to answer two main questions. How large is the climate impact of plant-based meat analogues compared to other foods? And which parts of the production are responsible for the largest emissions? And in this presentation, I will only focus on the findings related to the first question due to the limitations in time here. The literature review was performed 2 years ago and included scientifically published LCAs up to November 2021 that analyzed the climate impact of plant-based meat analogues. And to compare the results from different studies, we wanted to have comparable results, and therefore the choice of methods regarding functional unit and system boundaries were harmonized so that the results from all studies could be expressed in the same way as the climate impact at the industry gate, expressed both in relation to the product's weight, but also in relation to the protein content. We were quite selective about which products that were included. In the study, we were only interested in plant-based meat analogues that were developed specifically to mimic meat and where the main ingredients were protein isolate or concentrate. This means that products made from whole beans or vegetables, such as falafel, tofu, or vegetable patties were not included in this study. And either were foods where the main ingredients were algae, insects, dairy products, or eggs. So let's finally look at some results. First of all, I just want to give you a glimmer of the information compiled in this article, which was, for example, information about which products and raw materials that was studied, as well as nutritional information about the products, information on process technology and origin of production, and also information about the underlying methods and data used. When it then comes to the climate impact of plant-based meat analogues, we estimated that the medium impact of final products to 1.7 kilograms of carbon dioxide equivalents per kilogram of product, with a fourfold variation between the products studied. And here you can also see some results for climate impact expressed per 100 grams of protein, as well as results from the climate impact of protein isolates and concentrates, which were the main ingredients in these products. But what does these numbers really mean? To better understand what the results mean, we compared our results with data from previous research presented in the review analysis I mentioned in the beginning of this presentation. And for comparability between the studies and the data between the studies, our results were here harmonized with the system boundaries of the reviews. Still, comparisons between LCA data from different studies should always be made with caution. Nevertheless, the comparison do indicate that the climate impact of plant-based meat analogues per kilogram of product is lower than beef, cheese, and pork, but higher than most, but not all plant-based foods, such as beans and grains. The climate impact was also often, but not always, lower than chicken, fish, and eggs. And here, as you can see on the bar charts here, you can see an overlap between the minimum and maximum values of these product groups. On the next slide here, you can see that the conclusions were the same when the climate impact was expressed per protein content. But perhaps some of you are sitting here thinking now that it may be a little unfair to compare the climate impact of plant-based meat analogues, which are composite products made from several different ingredients with pure meat. So therefore, it is also interesting to compare the climate impact of protein isolates or concentrates with meat. And here you can see the results from our study again in comparison with the climate impact of meat from the review article by Clune et al. That represents available data globally, but also in comparison with data from Emma Moberg that represents the average climate impact from Swedish production. So when looking at this comparison, we should remember that the results from isolates and concentrates only come from 2 studies, and that the numbers are not 100% comparable due to slight differences in system boundaries. However, the comparison does indicate that the climate impact of isolates and concentrates is lower than beef. The median value is also lower than for pork and chicken, but here we again can see an overlap between the minimum and maximum values. So the results here will depend on what the production looks like. The title of this presentation was "How much do we know?" So I would also like to say a few words about data quality. In our review, a total of 9 LCA studies were including, so existing research was limited to relatively few studies. The literature review was conducted now almost 2 years ago, and we know that at least 3 more articles have been published since then, and that the climate impact in these studies were in the same order of magnitude as our results. When it comes to underlying data for calculating climate impact, we could also see that a large part of the studies based their calculations either on data from pilot scale production or secondary data from databases and scientific literature. In fact, only 2 out of the 9 studies included based the calculations of primary data from large-scale production. So here we really do need more knowledge. Finally, I want to say a few words about other environmental effects. In this study, we only focused on climate impact, but of course, there are many other important environmental perspectives to consider. A positive finding was that most of the studies included in the review analyzed more environmental effects than climate impact. The results showed that plant-based meat analogues generally have a lower impact than meat, also for environmental effects listed here on the slide. Although the results may vary depending on the products and environmental impact categories studied. And in the literature, the energy use of plant-based meat analogues is especially highlighted, as it can be in the same order of magnitude as some animal-based foods. So, in summary, what can we conclude from the results of this review? Well, the results indicate that plant-based meat analogues can be a climate-smart alternative to animal-based foods, especially if replacing red meat and cheese. And what the production looks like determines whether plant-based meat analogues also have a lower climate impact than other animal-based foods, such as chicken, fish, and eggs. The results also indicate that plant-based meat analogues usually have a higher climate impact compared to other less processed plant-based foods. The findings also highlight that current knowledge based on research is based on quite few studies with varying quality, where the need for more studies based on primary data from industrial production is especially highlighted. And finally, the large variation that we could see in climate impact between different products do show good opportunities for improvement in future production development. And here we can see that the choice of raw material, cultivation conditions, choice of energy source, and also protein separation technique are examples of factors affecting the overall climate impact. And with that, I thank you very much for your attention and open up for any questions.
Thanks very much, Eleanor. This was very, very interesting to see. And it's also interesting to conclude that we need more studies on this, definitely. So now we're going to have some input from the participating industry as well, which is Lantmännen and Orkla Foods. And I leave the word to Richard Gleserius from Orkla Foods, who will give a short introduction first.
All right. Hi, everybody. Richard Gleserius, Product Development Manager at Orkla Foods in Sweden. Try to give you a bit of a industry perspective for this project. And Orkla, we market and sell quite a lot of meat substitute and vegetarian products under different brands. And to start with, I would say, when we applied together with Lantmännen and Rice for this project, the meat substitute market was still booming. It was almost like a global race or competition to launch more and more meat-like products than the ones before. But things happen fast in this area of food business, and when we actually were granted the funds for the product, we had already begun to see a downturn in consumer demand for this type of product. Not catastrophic in any way, but still a dip in the curve after years and years of exponential growth. So from writing that application to actually starting the product, things had already started to change. So from a product development perspective, So instead of just looking at challenges and opportunities from a technical angle, we now had a discussion about bioavailability and a wish for more whole foods based products, and whole foods being, for example, whole pulses or grains, and not just the vegetable proteins. So the consumer demands have shifted along with the project. And on top of that, the economic situation in the world. So in a way, over the course of this 2-year project, the market has changed, and we have to pivot a bit in what to focus on in the project. So what we started with 2 years ago, we ended up with focusing more on fermented proteins than anything else, I would say, which has been very interesting. It's been an interesting journey. Personally, I think the whole project thinks so. We wish we would have had a bit more time to investigate a bit more when it comes to the fermentation, but at least it's a very promising beginning, and yeah, that leaves us asking for more. So let's see what's the next step will be. So. And with that, I leave the word to Karin Arkbåge at Lantmännen.
Thank you, and hello everyone. I'm Karin Arkbåge. I work at Central R&D for Lantmännen, and as you probably know, we offer a broad assortment of plant-based foods made of cereals and pulses. And this area of plant-based protein is of specific interest for us, both as ingredients and as consumer products, final products. We have an offer, but all from whole peas or cereal products onto more mashed products, looking like maybe a beef or minced meat or so, but also onto these more fiber-like, meat fiber-like products, extruded products. And I think it's very important for us all to consider the fact that this research area is very young compared to, for example, the area of milk or meat protein research, which has been ongoing for more than 100 years. So I think that's important. We need a lot more activity. There is a lot of research activity within this field today, but we still need more knowledge, and that is around functionality a lot, but also around nutrition, of course. And I agree with Rickard that it's been an interesting project, and we've learned a lot, and we also recognize that we need even more research within this field. And another thing that I think is important to consider is that also in a more immature market like the plant protein products market, first there are a lot of interest from consumers, and maybe the expectations are not that high, but when markets are maturing, then people are expecting more too. And I think maybe that is what we see now with a bit of a decline in the market, that now consumers are expecting more, and of course we want to live up to that and make the products, the next generation products, even better. And like meat is one of the pieces in that work. So yeah, I think that's my input.
Well, thanks to both of you. You mentioned that the market has been declining sort of the last year. Do you think you're expected to continue, or is it just a dip in the curve?
We expect and hope that it will turn around and actually increase the demand for this type of products again. Perhaps not the same products as we launched a couple of years ago. I think we're going to move more and more into the whole food based. I don't think that the soy based products are going to leave the market, but I think we need a wider variety of products to cater to the broader public.
Yeah, I think so too, and I don't think that one size fits all, but like earlier said, we need this variety of product concepts, and a maturing market will bring new and even more interesting concepts that consumers want to try. And we are in the green shift, and products will develop, I'm sure. And we already see that today, that with hybrid products and different concepts evolving.
A common question that we get in this area is that do you really need to mimic meat? Couldn't you just make a new product? What is your take on that?
I think for some consumers, you need to mimic meat to make the consumers try the vegetable products. But I think there are also a chunk on the market they don't want it to be like meat because that scares them away, or they prefer to eat meat because the meat as such tastes a little bit better, or at least more to what they expect the taste to be. So, and I think also that was the race a couple of years ago. Everyone wanted to make it as meat-like as possible. I think we're past that goal right now, at least as a single goal. I think we need to again have a wider portfolio of products.
And I think there's a lot of natural goodness in the plant-based assortment that we can lift and highlight even more in these products. And of course, a lot of natural goodness also in milk and dairy. So the question is also whether we will be, as consumers, better at cooking with more plant-based. That's more challenging, I think. But also that I think we will learn with time.
It's also more challenging from a production perspective, as you know that, I mean, the more protein, the easier to produce something that is meat-like. But it's also nice to see that we have shown that you can actually go down in protein content and still have a good product. So that goes in line with what you're saying. Do you see any specific new concepts that you think will come on the market? I mean, is it? I mean, so far it has been a lot of soy because soy is easy to produce and it's cheap, but will it shift to more Swedish? Will it shift to more other types of meats? Or do you have any ideas on this?
Do you want to start, Karin?
Yeah. I mean, we see different proteins like fungus-based. We also see hybrid products, a mix of meat and plant-based, which we saw like 20 years ago, but then the market wasn't ready, or the product wasn't good enough. But now we see that, and it's really interesting to see how those new concepts are accepted today. And also with new crops. I mean, we are curious and maybe we get more inspiration from other parts of the world too. That could be new for us here. To increase the variety is a good thing, I think.
Yeah, I think the Swedish origin of the raw material is, at least the consumers say that it's important if you're based in Sweden, of course. But right now, the consumers aren't really ready or willing to pay for that premium. So they say they want Swedish ingredients, but right now they're only ready to pay for soy-based products to be very like that. So I think hopefully we have a turnaround again with the economy, of course. So that will bring us back on track, I would say. to more Swedish ingredients and a wider variety of ingredients.
Yeah, and also building on that, I think it's important to highlight the cereal proteins, which are sometimes forgotten when we're talking about green protein shift, and we are considering legumes and a lot, but also our staple foods that we produce in India are important sources.
Yeah, and if you move away from products that need to be as meat-like as possible, then you have you have more ingredients to use. Like I said, you can use. It doesn't need to go past through an extruder to give you a meat-like structure. You can use the ingredients in other ways, and in that way you can use a wider variety of ingredients.
And if you could wish freely, what would you like as scientists to develop or focus on?
Yeah, I think functionality of different plant proteins is something that we need more knowledge on for different product concepts. Important.
Yeah, I would say the same. Yeah, even though we've just been talking about whole food products, I think still it's very interesting to learn more about functionality of proteins and how to use them and combine them. And also I think the fermentation part of it is also very interesting. What can we achieve with by fermenting the raw materials? Taste, texture, functionality. I think that there's a lot more to investigate and discover.
Mm. And we shouldn't, of course, forget about the nutritious part of the new food concepts that we really can absorb, and how it will affect our health. That's also something that we do. We do have research around in Sweden, and that both Orkla and Lantmännen and many others are part of too. So that's important.
Okay, so with that, it's just time to finish the seminar, and I would like to thank, of course, all the participants of the project for very great work, and Vinnova, Orkla, and Lantmännen for the support for the project. Without it, it wouldn't happen. And last but least, thanks to all the participants. It's great to see such a big audience, and we really appreciate that you're interested in the work we're doing. We hope to see you again. And with that, I just say thanks very much, and it's all for now.